Academic research is a habit of systematic questioning. It requires taking chaotic clinical observations and subjecting them to a rigorous process to formulate validated conclusions. We pass these observations through what we can call the methodological pipeline. To build this pipeline, you have to separate your tools from your methods. The library is a place to locate data. Statistics provide a way to summarize it. Neither is a research method in itself. This flowchart illustrates information gathering, extracting existing information, or collecting firsthand. Following the right branch, primary sources, is data generated for your study through observation, interviewing, and questionnaires. On the left, secondary sources is data already collected, historical documents, census publications, or earlier research. The origin of your data dictates its inherent biases. Whether you use primary observations or secondary census figures, you are introducing a baseline variance into your study that you must control throughout the rest of the pipeline. Because it is impossible to survey an entire population, we rely on sampling. By selecting a specific subset of respondents, we can estimate the parameters of the total population. The most robust approach is probability sampling. For a design to qualify as a probability sample, every single element in the study population must have an equal and mathematically independent chance of being selected. This diagram breaks down stratified sampling. Notice the split at step 6. Disproportionate sampling ignores stratum size, simply dividing sample size by the number of strata. Proportionate sampling calculates the exact proportion of each stratum, multiplying that fraction by the desired sample size. If your population elements are spread across wide geographical areas, you might use cluster sampling instead, grouping elements by location or institutional boundaries. Look at this map of Australia. Rather than randomly selecting students from across the entire continent, a researcher using cluster sampling would target specific, localized clusters, like the tightly grouped universities and technical colleges along the coastlines, and sample exclusively within those boundaries. We contrast these approaches with non-probability sampling. You use non-probability designs only when the total number of elements in a population is unknown or cannot be individually identified, relying on quotas or specific expert judgments instead of blind chance. Sampling is the structural foundation of your research. If you sample incorrectly here, every subsequent statistical test in the pipeline is mathematically compromised from the start. Once you have a valid sample, you can engage the quantitative logic engine. In this paradigm, statistics serve two distinct functions: describing your data and drawing inferences from it. Descriptive statistics summarize the general nature of your findings, identifying the point of central tendency and the degree of dispersion. Inferential statistics do the heavy lifting. They help you make decisions, determining if the differences between two groups are large enough to be attributed to an experimental intervention rather than an absolute fluke. However, inferential statistics are entirely dependent upon utilizing the correct measurement scale. Take an interval scale, like a thermometer measuring Celsius. When the temperature hits 0, it does not mean there is an absolute absence of heat. The 0 point is arbitrarily established. You can calculate means and standard deviations, but you cannot say 80 degrees is twice as warm as 40 degrees. Now replace that thermometer with a ratio scale, like a yardstick measuring a desk. Here, 0 represents an absolute point of origin. A measurement of 0 means there is no desk there at all, and a negative distance is a physical impossibility. This scale enables you to conduct virtually any inferential statistical analysis. The type of scale you use dictates the kind of study you can perform. Correlational studies simply investigate whether a mathematical association exists between variables. Explanatory studies attempt to clarify exactly why and how that relationship functions. Measurement scales are rigid mathematical gates. They dictate precisely which inferential tests you are legally allowed to run on your data. The ultimate goal of quantitative testing is establishing and challenging a hypothesis. It provides the checkpoint against which you test the findings your data reveals. Precise analytical tone. We begin with the null hypothesis, written as H0. This is the foundational assumption stipulating that there is no difference between two situations, groups, or outcomes. To test this, you rely on experimental design. The primary function of an experimental design is what researcher Fred Kurlinger calls the control of variance, isolating the effects of the independent variable while eliminating extraneous interference. The first step in this experimental flow requires dividing the study population and establishing a baseline metric for your dependent variable. This chart models the sequence. Once the baseline is set, different treatment modalities are introduced to the groups. After a set period, an after observation is carried out to ascertain exact changes in the dependent variable. This rigorous isolation is what separates true experimental flow from mere correlational observation, which only spots overlapping trends without proving cause. In quantitative research, we rarely prove that our theories are undeniably true. Instead, we rigidly prove that the null hypothesis is false, which isolates actual causation. Not all research fits neatly into numerical constraints. The qualitative ecosystem requires an entirely different approach to data. Because qualitative research explores areas where very little is known, formal hypotheses are rarely formulated or tested. Instead of predetermined checklists, data collection relies on unstructured methods. Researchers extract descriptive narratives, conduct in-depth oral histories, and identify emerging themes rather than subjecting answers to statistical equations. A prime example of this is the case study design. To qualify as a case study, the subject must be a bounded system. Whether you are studying an individual, an episode, or an entire town, you must treat the total study population as a single, holistic entity. In a quantitative model, we evaluate a study based on numerical validity. In a qualitative model, we measure credibility. Credibility is judged by respondent concordance. You take your findings back to the people who participated in your research. If the participants confirm that your analysis accurately reflects their experience and reality, the credibility of the study holds up. Qualitative research does not seek to measure mathematical variance. It seeks to map the complex topography of human experience that numbers simply cannot capture. To process either qualitative themes or quantitative metrics, we turn to computational environments. It is crucial to remember a key academic distinction. Utilizing statistical software is a tool, it is not a research method. A computer can calculate a correlation coefficient infinitely faster and more accurately than a person can. It will show you how closely variables associate. But it tells us nothing about why that association exists. Only the human mind can struggle with the underlying data to determine causation. Different tools serve distinct methodological needs. Excel is standard for basic descriptive measures, SPSS dominates the social sciences for structured inferential testing, and languages like R and Python allow for custom modeling and complex multi-layered data analysis. All of these tools operate on a strict garbage in, garbage out rule. They do not know if your measurement scales are correct or if your sampling frame was flawed. The software assumes your methodology is flawless. It will happily execute an invalid statistical test if you feed it badly sampled data. This brings us to the complete end-to-end methodological pipeline. A study flows sequentially. You determine your data collection method, apply a rigorous sampling frame, formulate a hypothesis, and finally subject the results to a software inference model. Each individual stage of this pipeline acts as a required filter against noise and variance. If a flaw enters at the top, it cascades through every subsequent layer until the final output is corrupted. Your assignment is to put this into practice. Select a software tool, extract a secondary dataset, apply a targeted, probability-based sampling frame to that data, and evaluate the output. True academic research is not about knowing which button to press in SPSS. It is about scientifically defending why you are allowed to press it.